Ashley. I'm going to be presenting the Heliocypha today. I'm going to be going over just a little bit about what this fish is, its phylogeny, morphology, and conservation. And then, time permitting, I'm also hoping to teach you guys a skill that you can put on your resume and say, technically, I know how to do this thing. So here is our Heliocypha, otherwise known as the Humpback Chub. You're probably thinking two things right now. First thing is, obviously this fish is beautiful. Second thing is, what the heck is that hump for? Well, don't worry, I will get into that. But first, let's go over the phylogeny. It's a bony fish, so Osteichthyes. It's a cypriniform, so it's a minnow. It resides within the stem branch named called the western clade. This clade encompasses fish that are typically endemic to the southwest of the United States. And then of course we have our Hilo lineage with Heliocypha within that. There are 19 species of the Gila, and because they are all endemic to the Colorado River Basin, there is a lot of hybridization and flip-flopping of those names. So even though this phylogeny is really clear, once you get into the actual genus, there's some weird things that can happen. But the Heliocypha is pretty well set in its phylogeny. So let's get into its anatomy and morphology. This first part here was taken from Fish Base, just a quick description. Over here, we have our Heliocypha again. You can see it's very long. It's actually known as these big river fish, so it gets pretty big if we let it. It has this very long caudal pedicle, a forked homocircle tail, and of course this big, large nuchal hump. The overall body form, as you can tell with all of these fishes that live in the Colorado River, is very similar, because the Colorado River has very high flows. Like, this is where the Grand Canyon is. This is where you're doing all of your whitewater rafting. So this body form of being slender and long helps you swim through those rapids. But that still doesn't answer the question of what is this hump for? So that is what these researchers did, Ports and Tyus. They set out to discover, like, okay, why do these things grow something that looks really energetically costly? Well, the Heliocypha here, along with A, who is Xyrauchium texanus, common name is the razorback sucker. They are both benthopelagic feeders. Bentho meaning the bottom of the riverbed, pelagic meaning swimming within the water column. So they swim around in these fast flows while eating at the bottom of the riverbed. So it was once thought that these humps helped push them down to the bottom of the riverbed. Rather than them having to take a lot of energy to swim down there and stay there, they thought, well, maybe the water flows over them and keeps them at the bottom. So to test this, these scientists did something we've seen in other papers that we've read this class. They built clay models, because you can't actually do experiments on these fish. They're very endangered. So they built clay models and then subjected these models to different types of flow. Very low water flow to extremely high flows that you would see in like a whitewater rapid system. And they calculated all sort of physics with this and discovered that no, these humps do not help them stay at the bottom. In fact, they do the opposite. Much like an airplane with wings, once you start adding things on to a very long and aerodynamic form, you will create lift, which is exactly what these humps do. They create lift, which actually makes it harder to feed at the bottom. So they had to come up with a second hypothesis, which brings us to this fish here. This is our North American pike minnow. It's the largest pike minnow in North America. They do get very big. And they prey on our razorback and our humpback chub, especially as juveniles. So they thought, like, okay, well, what is preventing these pike minnow from eating these big guys? Well, they measured the gape of the pike minnow and compared that to the depth of these fishes from dorsal to ventral side, and realized that the pike minnow's mouth on average, is too small to fit these fish with their big hump. So now it is believed that these humps developed out of a form of predator defense. So the moral of this story is actually body positivity. Humps are a great thing, and they can really protect you in certain situations. So that goes over a little bit of our morphology. Now let's talk about distribution. So as I said, these are endemic to the Colorado River Basin here. There's two parts, in upper and lower, and in red, those are our known breeding populations. Historically, all of this river system would have been red. But now, because of damming, they're very secluded. This right here is my area of interest for my research. This is the confluence between the Little Colorado River tributary and the main stem Colorado River, which is actually right here on this map. So these fish are known to breed in these tributaries, or spawn in these tributaries, which are warmer and have less flows. Then they can grow up and get big and make their way down to the main stem, where there's all of these whitewater rapids, where now they're big enough they can actually handle them. So that will bring us into conservation. Their biggest threat is the invasive species we all know and love, humans. We have, through damming and allocation of water for human use, have greatly decreased the flows which they rely on for spawning periodicity, and caused loss of habitat. So now throughout this river you have all of these whitewater rapids, but no floodplain, which is where actually these juveniles like to hang out. You don't want to be a tiny baby fish swimming around in these whitewater rapids. It's much better to be in a nice, cozy pool. So they were originally listed as endangered in 1973. They're still currently listed on the IUCN Red List. There's three main conservation programs listed here: the Upper Colorado River and Endangered Species Recovery Program, Glen Canyon Dam Adaptive Management Program, and the Lower Colorado River Multi-Species Conservation Program. So now I would like to talk about some techniques used to conserve these fishes, which brings me to my second paper. Otoliths reveal spawning ecology and early life history of sympastric catastonids. Now, you might be thinking, hold on, I thought we were talking about cyprinids. Why are you presenting a paper on catastonids? Well, that's because these authors here are my bosses. And this is the paper that I have used to inform my own research. So, using otoliths or ear stones, which grow daily annuli, you can back calculate when these desert fishes spawned and what their growth rate is. These can help inform, guide flow releases. Because, for example, if you know your fish are spawning in high flows, you want your dam to release water at that time of year, early May. But if you know your fish have spawned already and there's baby fish swimming around in your river in, let's say, June, that might not be the best time to release high flows. You're gonna wash all your baby fish away and be pretty detrimental to their ability to recruit into the juvenile stage. So this informed my research. So now let's talk about what I am currently doing. These are otoliths. This is a picture of them on a compound dissecting microscope. They're very small. And I have brought some slides that I have made where you can see how small they are because you can't see them. So inside of that crystal bond, there's a tiny ear stone. The bigger the fish get, the harder it is to count their otoliths, because the daily growth rings, although they're still occurring, they'll get so squished together that you won't be able to count them. They'll be too hard to decipher even in the most highest magnification. So I use really tiny larval fishes. You can pass this around too. You can see just how small these guys are. You cannot dissect them without a microscope. And this is so rewarding because this is polarized light, So blue light actually makes these guys glow. So you're digging through some brains, and all of a sudden you see these little gemstones light up, and you're like, I've got them. We look for very specific otoliths. There's 3. So we're looking for the lapilith. So this is under a compound microscope. This is about 200 times magnification. And you start seeing these rings very clearly. You get closer in magnification, and you start to be able to count these rings. Now, I'm going to tell you how to count these rings. Each ring that goes all the way around the center, or the primordia, is a daily growth increment. So, we have an otolith that looks a little bit like this. In the center, you're going to see a dark spot. That's the primordia. That's where the otolith is starting to grow. At about 100 times magnification, you're going to see these big, thick rings form all the way around that primordia, just like in this picture. See how these dark greens go all the way around? Well, once you start getting more into 400 times magnification, you'll see these rings break up. You'll see smaller and smaller ones. Those are what you're counting. Now the trick here is these fish can grow sub-daily increments. This happens when they're moving into a warm spot of water and they found some really tasty food for the afternoon. They're going to grow a sub-daily. Or the next day they move to a different type of water, find a different type of food. They're going to grow a sub-daily. The difference here is you'll see these rings sort of fold in and out like this. And where they'll even start and not go all the way around. You don't count those. So in order to count this otolith, I only want to look at the daily rings that are going all the way around this primordia. So I would count this as 1. Well, that blue one here goes all the way around. 2. That green one, it's not going all the way around my center. We don't count that one. This blue one is 3, 4, 5, 6. This fish is 6 days old. Pretty simple. It gets harder when you start getting closer and closer and closer into your magnification. And as I said, you'll notice that towards the end, as the fish gets older and older, they get closer and closer together, which is exactly why we use these juvenile fish. Once you get about 2 years old, they become so close together that even though they're there, they're impossible to count. Using this information, we can back calculate when that fish hatched and how old it is. This tells us exactly when spawning occurred, which we can then tell water managers, hey, spawning is occurring in May. Don't release your water here. There's all of these babies running around. They're gonna get swept away. Wait 1 month, and then you can release your water. This is a really practical way of supporting what little of these breeding populations are left. So this is my research that I'm currently working on. Hopefully I will have some results by next semester. And here are my citations for some of the information I presented today. And of course, lastly, are there any questions?